Our first speaker this afternoon is Dr. Andrew Veale. Andrew is a wildlife ecologist and a specialist in the ecology of mustelids and the application of genetics and conservation management. Andrew studied most introduced mammals in New Zealand, along with a range of birds, reptiles, insects, and other invertebrates. He's worked at Manaaki Whenua for 5 years, and in his infinite spare time, designs and makes telescopes, guitars, and guitar-like instruments that have been hybridized with others. Andrew's talk today will highlight several research projects on Waiheke Island and Taranaki, where they've used landscape genomics to inform and evaluate eradication programs of mustelids and possums. His research aims to understand their dispersal ecology and how successful control methods are at landscape scales. Thank you, Andrew.
Kia ora, everyone. Ko Andrew Veale, toka ingoa. So here's the best animal that you will see today. This is the stoat. And as anyone that has been to any talks of mine, so I worked on the stoat's genome and have a stoat genome tattoo. And that's the stoat for the stoat genome, and it is in Te Papa. It's in Auckland War Memorial Museum. And so it's really high quality, and hopefully we can use that for applied pest control. Now, how do we do that? This isn't genetic engineering. This is about understanding their populations. And one of the ways that I've been doing this is collaborating with Te Korowai o Waiheke to understand how their eradication is going. Now, that's their trapping grid. It approximates a 250 by 250 meter true grid across the island. And they're currently trying to check those every 3 days for large parts of the island, and a little bit longer intervals for areas that they know they don't often get stoats. So that's something that probably no one else can replicate. And back during my PhD, I suggested, because I did some genetics on the Waiheke stoats, that this was a really good place to do an eradication because the immigration rate is very low. And that means that if you remove them, they're not coming back quickly. So that's an amazing program. They've been going for a while now. If we look at the genetic diversity of stoats on Waiheke Island, you can see there used to be colours, there aren't anymore. Waiheke Island, the average, this is inbreeding on the y-axis. The average Waiheke stoat is as inbred as the most inbred animal on the mainland, which would be something like full sibling mating. So they are very inbred, very low population diversity. This is very good because it means, again, there's low immigration. If you then look at, so I've been getting every single animal that they catch and then doing genetics on them. And this is the genetic distance between any pairs of animals and the geographic distance. So in that yellow box, those are ones that are likely to be either parent offspring or full siblings. All right. And if we just look at those ones on the graph next to it, on the right, you see the distances between those animals. Now, there's obviously ones that are very close together. And so these are ones prior to dispersal. These are litters that have been caught near their parent, or their mother at least, and the other siblings. But then you have this interesting secondary peak at about 3.5K. And my interpretation of this is that the average stoat, when it disperses, it sort of runs around for about 3K and then settles, and then it's in its new place, if that is a niche that is not filled by another stoat, and then it will go into a trap. But you're also seeing that there are ones that go out to 12 kilometers. This is on an island that's about 20K wide. So they can do laps on Waiheke Island. But now let's look at what we can actually do when we have all of this genomic information from every individual. The red ones are ones that were a litter that was caught in 2020. And then that orange one in the center there, that is actually part of that litter. But she then was the mother of the yellow litter. And so those are her offspring, and you can see the dates. Now, there was actually one that went into the same trap as her 4 days after she went in, and that's one of her sons. But then these other ones are caught, as you see, it goes over time until March, April 2021. Now, that's a distance of 7.3 kilometers. So this is a juvenile stoat where mum has died early, and it's still gone across the most intense trapping regime in the country and hung out there for a while and gone 7.3 kilometers across it. That's a really scary thing. And this is the problem with trying to eradicate. You're trying to get every single animal, and many of these animals avoid, often for quite a long time. Now, if we didn't have the genetics, we wouldn't know that that's what was happening. We wouldn't know how far they're going. So this is a really useful tool in this very applied setting. Now, here's one the next year. So you have Dad. Dad is yellow. And then you have this litter, which is the red ones. So this was some results that I got very quickly for them. And mum was never caught. Now, I'm also getting the age of every stoat as much as I can through measurements of the skull or the baculum. And so we can try and get really, really fine scale. And it's like, okay, so mum was never caught. Therefore, she probably lives about there. And so I gave this talk to them and said, well, this is a good place to look. There is a female stoat that is avoiding things about there. And so they looked and they found a den using the dogs. So this is going from genetics of dead animals to finding dens. This is trying to really hone in how we can actually get to these last animals that avoid everything. This is last year's breeding for the animals that I currently have. And it's a horrible tangle. Now, the thing is that with a lot of this, you can't just stick it into a program. There isn't an off-the-shelf thing, go, who's mum, make a pedigree for me. It gets complex. Now, there are ones, so up in the top left of both screens, that the DNA just wasn't good enough. I'm going to try and get it again, but, so those ones I don't know. But the other ones, you can see these ones outside of the middle, they are connected to previous years. So it's okay, they were probably born in those years. But then there's this weird tangle in the middle. Now there's two litters. There's litter A and litter B, but they're linked to each other. That's complex. So what does that actually mean? Well, the thing is that both of the mothers were siblings and they mated with the same male, because that's what happens in stoats. The males come and mate with all of the females in the nest when they are young. So then the 205 is actually their brother, who was caught a year earlier. So this is the interpretation that we can start to get once you have all the data on all the animals. Now, what does that look like in reality? There's the two litters, okay? And something that I was really careful to do was I did all of the genetic analyses and I didn't plot anything on a map until I was like, I'm pretty sure I know what the truth is, and they look wonderful. So I think I was correct. Now, these other ones, these are ones that don't have siblings within the data set, the other colors, and then the gray ones are ones I don't currently have the genetics for. I'm getting that currently. But just let's have a look at some of these other ones. We've got females that were caught just before giving birth, lactating females that their litters were probably too young to disperse. And that could easily, if we take those three that I've highlighted in pink, that could easily be 30 other stoats that were taken out because mum died before they were independent. Really important that they got those. We also actually were able to live trap a male and bring him in, and my colleagues Patrick and Kyla have been looking at it, and it's a ninja stoat that avoids everything, very different. So we're getting all this information, and then I've been literally going through every single sample and going, where did this come from? Because we need to know that to try and refine this program. So you don't need to follow this, but I've been trying to go through literally every single one, trying to figure it all out. Now, last year, I would say there were between 2 and 4 litters, but there could have easily been 3 more. And males and females are avoiding traps for over a year. And they have a grid that means that this is not them not encountering the traps. They are encountering them. and avoiding. We know that this is a problem. In Ireland, there was a study recently that they had a camera inside a box and one outside the box, and 50% of stoats were only recorded on the camera outside the box, which is kind of a problem. You're having a trap inside a box, right? So it's really hard to get these last ones. Scent does appear to be important, and definitely when one family member's gone into a trap, another one will go in, and it's not just stoat scent, it's my brother's scent, you know? They're close to getting them. They're getting down to single digits of animals. But the problem with eradication is it's 100%. And 90% of a condom gets you to the same place as no condom. That's how biology works. We need to get rid of all of these ones, including the ones that avoid everything. And I believe that these results on Waiheke because we know that there's no immigration. Now, I think that they will get all of these individuals eventually through using thermal and eDNA and everything else. Eventually, they'll get there. They're getting down to low numbers, but right now it's a sustainable harvest despite that trapping regime. And so they're trying to step up. But on the mainland, this isn't a technique for eradication. Traps are not viable. And so we need to be having those conversations going, well, if we're going to eradicate, you have to have toxin. In Taranaki, we've been looking at how they actually move across, landscapes that aren't protected by 5 kilometers of water. And so we looked at these with GPSs and radio. That's really cool. But these are just a few animals. Now we want to know how the whole population is moving. I did, again, unfortunately, you don't get to see all the dots. There's lots of samples from Taranaki. They gave me about 600 dead animals. The stoats I got genetics from, so it's about even male and female, which means that they have enough traps to basically have a trap in every home range. But again, you'll have avoiders. But for the weasels, it's a high male bias. And that's because generally the threshold for trapping is about 100 grams. Female weasels are 50 grams, so they're not catching all the female weasels. That was an interesting result from the genetics. This is the movement of the few weasels that I was able to look at. And there is, I think, a 25-kilometer movement of a weasel. A little weasel running 25K. And that's what stoats do. So those are 40-kilometer dispersals of stoats across, again, one of the most trapped areas in the country. And that's if they went in a straight line. They probably didn't. They might have gone around the coast. They, had a fun time. Now, the longest dispersal of a stoat ever recorded, and this was an ear-tagged individual, was 65 kilometers. So let's just say by magic, I removed all stoats from Taranaki. How would I stop them getting back within a year? Have them not be in Waikato or horizons. That's the reality of stoat dispersal. It's very scary. And the good thing is that with all of this genetics, we can actually model it so that then you can go, well, how far is, 5% of them, 90% of them going? And this is the curve that we were able to fit. This is me and Tom Etherington working on this. And so as you can see, probably if you had a really good trapping grid out to 20 kilometers, most of them would be caught within that. But there's some that wouldn't. Now, that was all really depressing. I couldn't find out what landscape features affected stoats because probably none do, other than 5 kilometers of water or going over Mount Cook. But what about possums? Possums are not athletes. And therefore, they're actually affected by landscape features. Now, I love this map. Hopefully you will too. What I did was, from the 500 possums that they gave me, I looked at the level of inbreeding per possum. And then I mapped this spatially, which sounds really esoteric. But what it shows is that now green is low in breeding, red is high in breeding. You can see in the farmland, it goes more red. In the forest, it goes more green. This is because you have high populations with high connectivity in forest, and out in the farmland, you have low connectivity, so you're far more likely to mate with relatives. And what I found is the most likely scenario is that they come from the forest, They go out, they populate a small bush patch, and then it just inbreeds for a long time. Now, that basically also means that where is it easy to eradicate? Anywhere that has low population density and low connectivity, which is high in breeding. So this map is kind of how easy would it be to remove possums from this landscape using inbreeding values. Great stuff. Now, this is something called kin groups, which I made up. But basically, it's groups of related animals. So it's sort of like population structure. They're all more related if they're the same color. And you can start to see that so this dark red color, all the forest is kind of connected despite being far apart. But in the farmland, you have these little pockets of related individuals, which is what I was explaining. And generally, even this relatively small river, there's different colors on different sides, but occasionally there's one that crosses, often at the bridges or at the beach. So you can actually start to see what landscape features matter. And so Tom and I created a predictive model. And so let's just say, again, magic, there's no possums in the Kaitaki zero possum area. Within a year, where can possums come from with probabilities? This is the probability map for 1 year, and that's for 2 years. So then you can go, okay, if I want to have no immigration into this area with 95% certainty, I need to control out to this buffer zone. That's really important for management, right? You need to be able to go from, okay, possums move around the landscape like this. No, this is, okay, you've got 3 kilometers in this direction, do the control, you will have no immigrants. That's really good, useful information for managers. Something I did just for funsies, because they gave me a freezer full of possum heads. I found out that there's just a single gene for possum coat color. It's actually the same gene. It's called a Guti signaling protein. It's in horses and rabbits and everything else. So it's not that a black possum comes from Tasmania and a grey one comes from New South Wales. In New Zealand, often they're quite mixed and it's just one gene. Now, I am looking at other things now. This is a project with MPI for wallabies, both North Island and South Island. I've got about 300 wallabies that I process. I'm doing about 200 of those to look at how wallabies are connected. These are all my samples. And obviously, there have been some potential releases. So I'm looking at those and trying to find out where they are and if there are in situ populations outside of it. Also, goats on Banks Peninsula is a project that we're looking at soon for the eradications, and basically trying to go, well, how connected is all of this? So I think that this is a really useful tool in specific circumstances. And I'll be the first to tell you, if you come to me going, oh, I'd like to do a genetic study on this, I'll be the first to say, no, that's probably not useful to you if it's not, because it's useful in some circumstances for eradication. For other times, you just need to get on and spend that money on getting rid of them. But sometimes it can be really useful. And thank you. I have probably gone very quickly, but this is a lot of people's work. And any questions?
Yara, are you doing the whole genome or are you taking marker genes to do your...
So I'm using a process called GBS, which sequences about 1% of the genome, but it's the same 1% in all individuals. And then I get tens of thousands of markers. On Waiheke Island, it's about 30,000 because it's a highly inbred one. Taranaki, it's about 60,000. For the possums, it's about 50,000. So that's a lot of information. You slowly get less information as you go up to the millions of markers that you'd have for a whole genome.
The island, do you think that the inbreeding pressure could actually lead to some decline?
No, there's no evidence for it. I think that that population may have been colonized by a pregnant female a long time ago. It's very inbred. Doesn't seem to affect them at all. Strangely, some animals seem to be completely fine with that.
suggested this is most useful in an eradication, local extirpation. Do you want to expand on that? Is that mainly when it's useful, or are there other circumstances when it would be useful as well?
I think that for some control operations, it depends on where the recruitment is coming from. And so that's a question that could be very interesting. If you're doing control to low density, is it ones coming from external to that or from internal? If it's largely internal recruitment, then you don't need to know the connectivity. You need to get rid of those ones. And so it's that balance. And for different species, that will be a different thing. And sometimes we don't know. And so doing a genetic study will help us determine that. I think that most of the time, unless you're getting really low, recruitment will largely be from in situ breeding. And then you just need to try and get rid of those rather than worry about the connectivity. Connectivity does matter when you're getting down to very low numbers.